Gregor Mendel noticed that when he crossed purple-flowered pea plants with white-flowered pea plants, all of the offspring were purple. And he noticed similar things with other traits, so that tall pea plants crossed with short pea plants would give rise to all tall pea plants. And he came up with his law of dominance, where if there were different forms of an inherited trait, we would now refer to different alleles of one gene, that one gene would be dominant. So if you had offspring who inherited a dominant allele and a recessive allele, it would be one of the two alleles which would then give rise to the phenotype of the offspring. So that was one of Mendel's principles, his law of dominance. He then had as he tried to explain that and other things that he had noticed, his ratios, he came up with the law of segregation. In his law of segregation, he held that each gene was present in two copies in an individual. So, for example, these pea plants, they didn't just have one form of this trait, one allele, they had two. He was actually predicting the movement of chromosomes before anyone knew what chromosomes were. So he was saying that the leaves, the flowers, the pods, etc., these would be diploid, having two copies of each allele. In this case, this flower happened to have two copies of the recessive allele. This one had two copies of the dominant allele. But when they made sex cells for pollen and ova, that the pollen and ova would have one but not both of those alleles. That was his law of segregation, that these alleles then segregate so that the sex cells have one but not both. And we often represent this with Punnett squares, where we see that this individual, and right now we're only considering one trait, even though she might have two dominant alleles in her cheek cells, her bone cells, etc. When she makes gametes, her gametes will have only one, but not both. His sperm will have this one or this one, but not both. So these alleles segregate in the offspring. And so here we see a Punnett square. This is perhaps even more noticeable when we look at a heterozygote. So a heterozygote would have then half of their sex cells. So although her cheek cells have a dominant and a recessive allele, her bone cells have a dominant and a recessive allele. Her ova will have one or the other, but not both. So half of her ova will carry the dominant allele, half of her ova will carry the recessive allele, and the same for his sperm. Half the sperm will carry the dominant allele, half the recessive allele. And then when they randomly fuse, this is what gives us our ratios. So those are two of Mendel's three laws. Mendel then developed a third principle because he asked what would happen if now we looked at the inheritance of two traits at the same time. Okay, and so let's just say, for example, we're looking at a gene which controls whether you or not you can roll your tongue. So having a dominant allele would mean you can roll your tongue. Being homozygous recessive would mean that you can't. Let's say that we then look at a second trait. In the previous example, Mendel only looked at one trait, where we look at your earlobes. If you have a dominant allele, then part of your earlobe hangs free. If you have a recessive allele, then no part of your earlobe hangs free. Well, let's now look at this individual who is homozygous dominant for two separate traits. Okay. Now, his alleles do segregate, all right, because if this is, say, the dominant allele he got from his mother, this is the dominant allele he got from his father. This is the dominant allele for the second gene he got from his mother. This is the dominant allele for the second gene he got from his father. Then his alleles are segregating because half of his sperm are getting the dominant allele for tongue rolling that he got from his mother, half are getting the dominant allele for tongue rolling he got from his father. But for our purposes, it doesn't matter. All of his sperm then would have two dominant alleles. And so if he only has one form of the A allele, then even though he has two copies, one from each parent, all of his sperm will get that copy. If he only has one copy of the B allele, the same thing. If this woman is homozygous recessive for these two traits, then all of her ova will receive an A allele and a B allele. Now, I just want to repeat that because sometimes my students will get that confused, where they will say, this ovum is little a, little a, little a, little a, little b, little b, little b, little b. No, each gamete is getting an A allele and a B allele. Our sex cells have all of our genes. So we don't have, some gametes with the A gene and some gametes with the B gene. But the law of segregation means that the gametes will not be getting both of these alleles, one she got from her mother, one she got from her father, only one of them. All right. Now, even though half of her ova will get this allele and half of her ova will get that allele, for our intents and purposes, it's the same because she's homozygous. There's no difference at this point for these for those different alleles. And so what we then notice is if you are homozygous for two separate genes, then all of your gametes then are the same, where you have this individual who has two dominant alleles. He then has all of his sperm with a dominant allele for the first gene, a dominant allele for the second gene. She has recessive alleles. And once again, I'm giving different examples here. It doesn't have to be, tongue rolling and earlobes. It could be cystic fibrosis and phenylketonuria. This would be the same. Now, just before we go into independent assortment, we just, have to master, this process of what gametes would they make. So let's imagine now that this individual is homozygous for one of the two genes, whatever that gene may be, but heterozygous for the other. Now, half of his sperm will get this B allele he got from his father, half of his sperm will get this B allele from his mother. But since they're identical, we don't need to distinguish between those. But we do need to distinguish between this dominant allele and this recessive allele. So half of his sperm, all right, are getting a dominant allele, sometimes with this B that he got from his mother, sometimes with that B he got from his father. The other half of his sperm are getting the recessive allele for the first gene. And then the same thing would be observed if this woman were to make gametes. Notice she's homozygous for the A gene, but heterozygous for the B gene. So both of them make two types of gametes. In the previous example, if you're homozygous for both, you only make one kind of gamete. These individuals are heterozygous for one of the genes, so they're making two kinds of gametes. All of their gametes have, as in this individual's case, a dominant allele for the second gene, but half of his gametes have a dominant allele for the first, half have a recessive allele from the first. All of her gametes have a recessive allele for the first gene, but half of her gametes have a dominant allele for the second gene, half of her gametes have a recessive allele for the second gene. Now, once again, the beauty of this is that that could represent any pairs of genes once again. Now, if an individual is heterozygous for both of these genes, then sometimes this allele is associated with that allele or with that allele. This allele could be associated with that allele or that allele. And so then there are four possible combinations. This first allele for the first gene could be associated with the first allele for the second gene, or the second allele for the second gene. So this one or this one. And then this second allele for the first gene could be associated with the first allele for the second gene, or the second allele for the second gene. So this one and this one. So there are 4 different types of gametes: big A, big B, big A, little b, little a, big B, little a, little b. And then the same would apply for her as well. So if someone is a double heterozygote, if they are heterozygous for 2 different genes, then there are 4 different combinations of gametes that they then could make. All right. Now, Mendel was working with plants. We often use the term hybrid for plants. And so then these individuals would be dihybrids. They would be heterozygous then for 2 different genes. This is just something we have to master before we then go to our next question. In our next question, what would we then observe? All right, let's say that 2 individuals who are heterozygous, then have children. What would we observe? Well, remember that the original Punnett square that I showed you, there were 2 types of gametes. Say maybe one had a big T, one had a little t. Well, here, if someone is a dihybrid that makes 4 possible gametes, then we have to have now 4 columns here. If this individual makes 4 possible gametes, then we have to have 4 rows here. All right, so in the Punnett square, which just had 4 blocks, it was 4 blocks because each of them made 2 types of gametes. And so we had 2 columns, 2 rows. That could be the same here as well. So for example, if someone is homozygous for one gene and heterozygous for the other, once again, they make 2 kinds of gametes. And so we could represent it with only 4 blocks as well. But if they are dihybrid, each individual then has 4 different kinds of gametes. Some of the gametes will have dominant alleles for both genes. Some of the gametes will have a dominant allele for the first gene, a recessive allele for the second gene. Some will have a recessive for the first, dominant for the second. Some will have a recessive for the first and a recessive for the second. So a woman who is a heterozygote for 2 separate genes, we would then realize she would make these 4 different kinds of gametes. If a man was heterozygous for 2 different genes, he would make 4 different kinds of sperm. And then we would simply ask the question, what would happen if this sperm fertilized this ovum? If this sperm fertilized this ovum? And then we would just, if the sperm has a dominant A and a recessive B, and that ova has 2 dominant alleles, then this individual would be big A, big B, little b. And so we would just then go through the various combinations, combining whatever allele was present, or whatever 2 alleles were present in the sperm with whatever 2 alleles were present in the ovum. So this is what we would observe in a dihybrid cross. Now, in the original cross, we were then concerned with not only the genotype, but then the phenotype. All right. So let's go through an example with flies where we're looking at a phenotype. Now, it turns out there are these 2 genes, brown and scarlet. And if you have the wild type dominant alleles for each, then the fly has its normal wild type red eyes. So let's just say that the A gene is the brown gene, the B gene is the scarlet gene. Once again, just A and B are representing first and second genes. So this female fly is homozygous dominant for both, this male fly is homozygous recessive for both and has white eyes. There are different ways that flies can have white eyes, but in this example, if you don't make red pigment and you don't make brown pigment, since they're the only two pigments, then you have white eyes. And so if we have a cross where a homozygous female who is homozygous for two different genes, so she has two dominant alleles, we ask what ova would she make? Well, all of her ova will carry an A gene and a B gene, all right? But because she was homozygous, she only, essentially makes gametes that have a dominant allele for the A gene and a dominant allele for the B gene. If this male is homozygous recessive for two genes, all of his sperm will carry two recessive alleles for the A gene and the B gene. So all of the gametes have both A and B alleles. And because these individuals were homozygous for both, then the gametes are then, essentially the same coming from the male, and the same coming from the female. If we then ask, all right, well, then what happens? Well, if a sperm, which carries a recessive A and a recessive B, fuses with an ovum, which has a dominant A and a dominant B, then the offspring have dominant and recessive alleles for both A and B. They are doubly heterozygous. They are dihybrids. Right? And so here we have flies who are heterozygous for two different genes, A and B. Now, since these alleles are the dominant ones, then they would all have red eyes. All right. So in this cross, the double heterozygotes, the dihybrids, they have the dominant phenotype. Now, as we had just gone through, each of them would make four different kinds of gametes. So the female, because she's doubly heterozygous, would make four kinds of gametes. Two dominant alleles, a dominant and a recessive, dominant for the first, recessive for the second, recessive for the first, dominant for the second, recessive for both. The male, being doubly heterozygous, would make four kinds of sperm, and then we would get this enlarged Punnett square with 16 possible outcomes. Now, we could go through the genotype ratio, but I think it's preferable to focus on the phenotype ratio. 9 of the 16 combinations have at least one dominant allele for the A gene and at least one dominant allele for the B gene. So there is at least one dominant allele for each of the two genes, so that 9 of the 16 combinations would then have red eyes, being doubly heterozygous. Then 3 of the combinations would have a dominant allele for the first gene, but a recessive allele for the second gene. All right. They would have brown eyes. Here we have 3 combinations where they have they're homozygous recessive for the first gene and have at least one dominant allele for the second gene. They would have scarlet eyes. And then 1/16 of the time, a sperm which has 2 recessive alleles would fuse with an ovum with 2 recessive alleles. And without any dominant alleles being able to transport these pigments, then the flies would have white alleles. So if you were counting the F2 generation, you would notice a 9-to-3 to 3-to-1 ratio. So that on average, 9/16 of the flies that you count having 2 dominant alleles, or at least a dominant allele for the A gene and a dominant allele for the B gene, would then have brick red eyes. 3/16 on average would have brown eyes. 3/16 on average would have scarlet eyes, and 3/16 being homozygous recessive for both would have white eyes. So this is a dihybrid cross, and it only works if the genes are assorting independently. So if that's what you observe, if you observe that um, for these two traits, you end up getting a 9-to-3 to 3-to-1 ratio, then that would indicate independent assortment. Now, as I'll pursue later, you don't always know. So for example, let's say that here we have flies with an ebony body and sepia eyes. Now, I realize that some people might have been a little confused that even though we were dealing with two genes in the previous example, they both affected eye color. Two different genes could affect obviously two different parts of the body. So here we have one gene that's affecting eye color, one gene that's affecting body color. And we want to ask, how are they inherited? Well, what Mendel observed and then what he would predict, it doesn't always work for important reasons, which I'll get to in a second, is what's called independent assortment. So if sepia ebony was assorting independently, then what we would observe is that this female having two dominant alleles would make gametes, which have a dominant A and a dominant B, one for the sepia gene, one for the ebony gene. If this male were homozygous recessive for both of those genes, all of his sperm would carry a recessive allele for the sepia gene, a recessive allele for the ebony gene. And then as such, all of their offspring would then be double heterozygotes for both the first gene and the second gene. Now, if the wild type alleles are the dominant alleles in this case, being dominant for the eye color sepia and being dominant for the body color of ebony, then in this case, then all of the F1 offspring being double heterozygotes, they would all have red eyes and tan body colors. Being double heterozygotes, they would make four kinds of gametes. So the females would make these four types of ova, the males would make these four types of sperm, and then we would expect to have then 16 combinations of alleles. 9/16 would have the dominant condition for the first trait and the dominant condition for the second trait. So they would have red eyes and a tan body. 3 of the possible conditions would be dominant for the first trait, say they would have tan body, but recessive for the second, say they would have sepia eyes. 3/16 would be recessive for the first trait, but dominant for the second trait. So they would have ebony bodies, but red eyes. And then 1/16 would be recessive for both. So once again, if we were to count the offspring, we would expect a 9-to-3 to 3-to-1 ratio in these phenotype categories. Now, we might not get that, and specifically, we might not get that for sepia ebony. But before I explain why, let me go to this explanation. Mendel didn't know what chromosomes were, but he predicted their movement. So once again, that's just a testament to the power of his conclusions. Now, let's just look at two different human chromosomes. Let's say the big chromosome is chromosome 1. Let's say the blue one's the one you got from your mother, the pink one's the one you got from your, obviously the blue one's the one you got from your father, the pink one's you got from your mother, obviously it wouldn't matter. And then the smaller one, number 7, let's say the blue one is the chromosome 7 you got from your father, and the chromosome 7 you got from your mother. Now, notice that the chromosome 1s have these red genes, but there's a light red allele and there's a dark red allele. Notice that the chromosome 7s have yellow genes, but there's a bright yellow allele and then a duller yellow allele. So what types of sex cells would someone make? Well, Mendel said these assort independently. They don't hang out in a group. All right. So what you're about to see, this could happen, but then so could something else. And so here that as they line up in prophase 1 going into metaphase 1 of meiosis, we could line up this way, where the dark red allele is on the left, but the light yellow allele is on the left. We could go out that way. So notice that this gamete will now have chromosome 1 that this individual got from their father, but chromosome 7 that they got from their mother. Notice that the blue chromosomes, they didn't hang out in a gang that are all traveling together. And if the blue chromosomes don't hang out in a gang that travels together, then the alleles that they carry don't hang out in a gang that travel together. And so you did not get the two dark alleles in the same gametes that time. But notice that they're not hanging out in a gang, they're assorting independently. So the next time that an individual makes gametes, it might turn out that the two blue alleles line up on the same side of the metaphase plate. I'm sorry, the two blue chromosomes with the dark alleles, and the pink chromosomes line up on the left with their lighter alleles. And so when someone makes sex cells, this was, you could have a big A and a big B, or you could have a big A and a little b. This is the explanation. You could have a maternal chromosome 1, you could have a pink chromosome 1 with its bright red allele. And as your second one, you could have a pink chromosome 7 with its bright yellow, or you could have a blue chromosome 7 with its dark yellow. So notice that the chromosomes don't look at where the other one is going and then decide, oh, I'm going to go left because you went left. They're assorting independently. And because the chromosomes are assorting independently, the alleles which are on them assort independently. So if we were to now have, the big A, little a, so if the A gene was on one chromosome, let's say chromosome 1, and the B gene was on a different chromosome, let's say chromosome 7. The reason that we were getting random combinations of big A, big B, big A, little b, et cetera, was because these chromosomes assort separately. They're not physically attached. So Mendel noticed that the P's that got the dominant A trait didn't always get the dominant B trait. Those traits were assorting independently. That 9:3 to 3:1 ratio implies that there is the random fusion of gametes. So when someone makes gametes, you could get a scenario where both dominant alleles are in one gamete and both recessive alleles are in another gamete. But you could just as easily then get the opposite a scenario where a gamete then has a dominant allele of one versus another. So notice here that once again, if we were to just say, the blue chromosomes represent the chromosomes from one parent, the pink chromosomes of the opposite parent. At the metaphase plate, they could all line up on the same side, in which case the sex cells would have whatever alleles that the father had in one gamete, and then all of the alleles that the mother had had in a different gamete as they separate. All right. So here, if this sperm were to fertilize an ovum, then it would have all of the originally paternal alleles, while if this sperm fertilized an ovum, it would have all of the alleles that that man inherited from his mother. But obviously, chromosomes don't have to line up that way. All right, so instead, they could line up in different combinations so that the sex cells would then have combinations of maternal and paternal alleles. All right. So before I go then to the exceptions, what Mendel observed was that the alleles for one trait don't always travel with alleles for the second trait. So if the female fly in the first generation had dominant A and dominant B alleles, they don't always travel together. All right. If the dominant A and the dominant B always travel together, then the only offspring we would have in the grandchildren generation, the F2, would be red-eyed flies, which have the big A and big B that always travel together. And the white-eyed flies, those would be the only combinations. But here we're getting brown, where you have the mutant alleles for one gene, but not the other, or scarlet, where you have mutant alleles for the second gene, but not the first. And so Mendel observed independent assortment of chromosomes, and it is one of his three principles. Now, do traits always assort independently? No, they do not. So I'd like to end with this, but this then should then be something that we would return to, because very often you do a test to see if traits are assorting independently. And so, for example, let's say you're looking at two fly mutations known as black and vestigial, black body and vestigial wings. And you say, are they assorting independently? And if you do a cross where you have a parent which is homozygous for both, say, wild type traits, a parent which is homozygous for both mutant traits, you have the next generation, the F1, which are double heterozygotes. And then when you look at the F2 generation, you see a 9 to 3 to 3 to 1 ratio of 9/16 of the flies have the wild type trait for both traits. 3/16 are mutant for the first trait, but wild type for the second. So they're mutant for body type, but normal for wings. 3/16 are mutant for the second trait, wings, but normal for body type color. And then 1/16 is mutant for both. If you get this trait, you say, oh, it's a 9 to 3 to 3 to 1 ratio. These genes are associating independently. But what happens if you don't get a 9 to 3 to 3 to 1 ratio? Very often, you're so excited. That's what you were hoping for. So you don't have to get a 9 to 3 to 3 to 1 ratio. And you wouldn't if the genes were linked. See, they assort independently if they're on different chromosomes. But let's say that they were stuck to each other. So let's say that the big A always travels with the big B, and the little a always travels with the little b. Then you would only make two types of gametes. All right, the gametes would either have dominant alleles in both or recessive alleles in both, in which case then your offspring in the F2 generation, we wouldn't see a 9 to 3 to 3 to 1 ratio. You would just see a 3 to 1 ratio, where you either say have red eyes or white eyes, none of this brown or scarlet, combinations. So if they were completely linked, these two genes always traveled together, then you would get not a 9 to 3 to 3 to 1 ratio, but a 3 to 1 ratio. What happens if they're linked a little bit? All right, so that they're linked, but that crossing over can separate them. Now I'd like to end with this because I'd like to then revisit, this a little bit later as a form of mapping, and it is very important, but I'd like to just mention this. So if they're linked but not completely linked, you would get lots of flies which are dominant for both, lots of flies which are mutant for both. But look, here you can see ebony flies that don't have vestigial wings, or vestigial flies which don't have ebony wings. You would get some of these intermediate combinations. Now, once again, this becomes a very important point later on, but here's, I guess, big picture. If you don't know where a gene is, then there's not a whole lot you can do. You can say somewhere there's a gene that causes this trait in flies, or this trait in humans. Here's a human disease. We don't know what gene causes it. There's a gene out there somewhere that causes it. We just don't know where. What you often then do is say, all right, let's take this gene that we don't know and this gene that we do know, and we know where it is. And let's see if they assort independently. If the answer is yes, they assort independently, then you say, all right, they're on different chromosomes. But if you don't get a 9 to 3 to 3 to 1 ratio, all right, that's not a problem. It means you've learned something. You say, the genes are linked. So here, will the bright red and the bright yellow alleles assort independently? Will the dark red and the dark yellow alleles assort independently? No. Why? Because they're stuck to each other. They're on the same chromosome. They're linked. If you do an experiment with flies or with humans, or, and you say, I'm not getting that ratio I'm expecting for independent assortment, the genes are linked, you say, I know where the gene is. And that is so exciting, because once you know where the gene is, you could then try to physically, it's like it's right here. Then you could sequence it. Once you sequence it, you say, oh, this is a membrane protein. Maybe this, maybe this medication, which works on membrane proteins of this gene family, would work on this disease. Let's try that. Let's do a genetic test. Now that we know where it is, let's screen patients and see, do you have different forms of the DNA that's at this spot? Oh, this seems to be running in the family, and this member of the family has a potential to pass it on to their offspring, but not this one. Or this person would benefit now from early screening. This person wouldn't. We might save this person's life. This procedure, which would occur, say, after age 40 or after age 50, let's do it in their 20s. We might catch the problem early. Once you know where a gene is, there's so much you can do with it. Before you know where a gene is, not so much. And so very often you ask the question, are these two genes independently assorting? Now, sometimes they are, and you're getting a 9-to-3 to 3-to-1 ratio, because they're either A, on different chromosomes, or B, on the same chromosome, but really far apart from each other, because if they're really far apart, they kind of like assort independently. Anyway, for reasons which we'll get into later. But if they're not assorting independently, you can say, I just found my gene, and that might put you on the road to sequencing the gene, figuring out what kind of protein the gene does, screening people for a disease, which might save lives, developing, medications, which, address that type of protein. So, this could be a huge, huge step. And so, once again, one of Mendel's 3 rules was independent assortment. When genes are on different chromosomes, they do assort independently. When they're on the same chromosome, they don't. Now, sometimes, and once again, this is an upcoming topic, crossing over can separate them, all right? So, the dark alleles don't have to stay glued together for all eternity, nor the light alleles. When we undergo meiosis in prophase 1, there is crossing over that can shuffle this a little bit, but that's a far bigger topic with far more important applications. So I'd like to pause there and then simply sum up. Mendel had 3 principles. The principle of segregation, of dominance, and of independent assortment. If genes are assorting independently, then they're on different chromosomes. And that means that the big A allele doesn't always travel with the big B allele. If they did, then this fly's offspring would then always have both dominant alleles and always have red eyes. But it turns out that the A can travel separately. Notice that not every big A is with a big B. Note that not only every little a is with a little b. So the A and the B alleles are assorting independently into gametes. And the way that we know this is the ratio of phenotypes, which we notice in this dihybrid cross, where we get a 9-to-3 to 3-to-1 ratio. That's what we would expect if these are randomly assorting, because they are independently assorting. And once again, there's a statistical argument here, and that will be in a separate